Welcome to Open School. Today I am discussing Competency Biochemistry 1.1, the functional organization of cell and subcellular components. As we know, this is the human cell. I will be going into details of some of this. So, the conceptual framework, at the end of this, you'll be able to understand these things. That is about the cell as a unit of life, the molecular composition, plasma membrane, cytoskeleton, mitochondria, endoplasmic reticulum and Golgi apparatus, lysosomes and peroxisomes, and nucleus. This is only just a brief introduction to these things. Many questions are asked in the examination, and these are the fundamentals that you should know. First question: explain the fluid mosaic model of the cell membrane. As we know, this is the plasma membrane. This consists of a phospholipid bilayer. So here you can see the phospholipid bilayer with the hydrophilic heads outward and hydrophobic tails inward. So this portion, this is the outside is the hydrophilic and the inward portion is hydrophobic. That is, you should remember. Integral parts are the peripheral proteins which float in the lipid matrix. You can see that as protein, transmembrane proteins are floating. So the cholesterol modulates the fluidity. So here you can see the cholesterol, and this is the thing that gives the fluidity to the membrane. Glycolipids and glycoproteins form the glycocalyx for the cell recognition. So you can see the glycolipids here. They give identity for cell recognition. So the model emphasizes that the membranes are not rigid but dynamic self-healing structures. So this is the fluid mosaic model. So there are integral proteins are there, or proteins are floating in the sea of lipids. Again, remember the lipids hydrophobic inside and hydrophilic outside, and there are glycolipids which gives recognition to the cells. Details you will learn later. One of the important structures is mitochondria. Mitochondria is the powerhouse of the cell. It's a double membrane. You can see there is a one membrane outside and there is another membrane which is forming the cristae. It's a double membrane organelle, permeable to small molecules. The inner membrane is the one which is folded into cristae. You can see they are folded into the cristae, housing electrons transport chain and ATP synthesis complexes. So that's why it is called the powerhouse of the cell. The matrix contains mitochondrial DNA, ribosomes, enzymes for the tricyclic TCA cycle, fatty acid oxidation. Those main functions are the ATP production, beta oxidation of the fatty acids, apoptosis regulation, that is by release of enzymes like cytochrome, and calcium buffering. Details will be in subsequent lectures. So, as mentioned, mitochondria is the powerhouse. So the oxidation metabolism provides 90% of the cellular ATP. And again, remember, mitochondrial genes are inherited maternally, and there are many diseases which are inherited through mitochondria. So you should be able to draw the picture of this. There is mainly the double membrane, and you can see this is outer membrane permeable to many small particles. Inner membrane is folded into cristae which contain ribosomes. These enzymes. So electron transport chain is going. So then there is a mitochondrial DNA. As mentioned, mitochondrial DNA is inherited from the mother. What happens if mitochondrial oxidative phosphorylation is uncoupled? ATP synthesis stops though the electrons transport continues and energy is released as heat. So that's one point here. Remember, that is when the mitochondrial oxidative phosphorylation is uncoupled, ATP production lags, but the electron transport continues and the energy is released as heat. So what is the importance of mitochondrial function? Mitochondria is the powerhouse. Energy, 90% the energy is obtained through the mitochondrial beta oxidation and other things, and it produces dysfunction in all the organs. It can produce disorders of eyes, heart, pancreas, skeletal muscle, kidneys, so all the organs. It can produce various diseases. So there is a separate some or one or two diseases are important. One is for the it produces myopathies and lactic acidosis, stroke-like symptoms. So they are called MELAS and MERF, about which you will study later. So remember, mitochondrial function is essential for all the organs. Now comes the lysosomes and peroxisomes. What are lysosomes? Lysosomes are membrane vesicles containing hydrolytic enzymes which are active at acidic peaks. So they degrades macromolecules, autophagy and recycling. So the enzyme deficiency leads on to diseases called lysosomal storage diseases. There's two examples you just remember. One is a Tay-Sachs disease and Gaucher's disease. These are lysosomal storage disease due to dysfunction or disorders of the lysosomal enzymes. Similarly, there are structures called peroxisomes which contain oxidases and catalases. They participate in the beta oxidation of very long chain fatty acids and detoxification of hydrogen peroxide, and this also leads on to various diseases called peroxisomes diseases. I mean, remember, it is oxidases and catalases. They are important for metabolism of the very long chain fatty acids. So these are number of diseases produced due to the lysosomal storage disease. It again affects, as these are present in various organs, multiple disorders. You can get disorders of heart, brain, kidneys, abnormal facies, skin, and whatever organ or any organ can be affected. So as mentioned again, the peroxisomes in humans also leads on to disorders of metabolism of very long chain fatty acids. That leads on to various disorders, and again this will be a separate topic, peroxisomal. So it has a oxidases and catalases, whereas this lysosomal storage, it is the destruction of the macromolecules. So disorders of this leads on to what is called the lysosomal storage diseases. Examples such as Tay-Sachs disease and Gaucher's. So now we go into the synthesis of protein. Which organelles are involved in protein traffic? So remember, this is the rough endoplasmic reticulum surrounding the nucleus, and it is rough because of the ribosomes. So the protein trafficking occurs through the rough endoplasmic reticulum, then goes to the Golgi apparatus. The Golgi apparatus modifies the proteins, then goes to the secretory vesicles. So here you can see the secretory vesicles. Then from the secretory vesicles it goes to the plasma membrane or to the lysosomes. Its enzymes, it goes to the lysosomes. So this is the protein traffic. So rough endoplasmic reticulum to the Golgi apparatus, to the secretory vesicles, and then to the lysosomes. So again you can see the endoplasmic reticulum. A small part is enlarged. This is the lumen of the rough endoplasmic reticulum. So proteins are inside, then it is produced into the vesicles. So what is the function of the smooth endoplasmic reticulum? Smooth endoplasmic reticulum lacks ribosomes. It synthesizes lipids, phospholipids, and steroid hormones, and it detoxifies many drugs. So the difference between endoplasmic reticulum, rough and smooth, rough contains the ribosomes, studded with the ribosomes. That is why it is called rough, and it is a cytosynthesis of secretory and membrane proteins. Hope it is clear to you. What are the disorders of Golgi processing? Golgi apparatus process the proteins. So the disorders can lead on to many disorders of, like example, disorders of glycosylation. Again, the Golgi dysfunction can lead on to disorders of central nervous system, lung, produce osteoporosis, gastric cancer, colon cancer. So many diseases can be due to the disorders of the Golgi apparatus. So Golgi apparatus, it is a stacking of the proteins or modification of the proteins is in the Golgi apparatus. Why is compartmentalization is crucial for cellular function? It separates incompatible reactions. For example, oxidative reductive process increases efficiency of concentration of enzymes. For example, the lysosomes, peroxisomes, allows regulation of the metabolism. So the fatty acid synthesis occurs in the cytosol, whereas beta oxidation occurs in mitochondria. So that is why the functions are compartmentalized. So it is a few MCQs based on this. The main site of beta oxidation of fatty acid is, you write down the answer. What is the answer? The answer is mitochondria. Beta oxidation is a powerhouse, 90% of the energy. Next question, the smooth endoplasmic reticulum is abundant in cells involved in. So we have mentioned the rough endoplasmic reticulum is concerned with the protein synthesis, whereas the smooth endoplasmic reticulum, the correct answer is mainly with the steroid synthesis. Damage to lysosomes primarily leads on to, but lysosomes, it contains catalytic enzymes. So it is a autophagy, so it is a cellular lysis. Answer is the B. So there can be some diagram based questions. You should be able to draw the electron micrographic picture. All of you stop the video and try to draw this picture and identify the structures. So this is summary of the MCQ question. So cytobeta oxidation, it is mitochondria. Enzymes of beta oxidations are in the mitochondrial matrix. Smooth endoplasmic reticulum function, mainly the steroid synthesis, abundant steroid producing cells. Lysosomal damage effect, cellular autolysis. Release of hydrolytic enzymes causes cell digestion, autophagy. The organelle with the circular DNA. What is a cellular substrate with the DNA? It is mitochondria. As I mentioned, mitochondria genes are maternally inherited. Component responsible for membrane fluidity. So the fluid mosaic model, the cholesterol is the one which gives on fluidity. So what is the reflection? Why is the cell called fundamental unit of life? The aspect of reflection is because every physiological and pathological process originates from organized molecular interactions within cells. Disturbance in cellular organization lead to diseases. So this completes the biochemistry, this CBME based topic. This is a conceptual framework. You can go through this. Stay tuned for further videos. Thank you.